principles of chemistry, what you need to navigate this course and your other life science courses as well. Okay, so put that in there. Yeah. So we got that. Ready to go. Okay. So when we talk about chemistry, one of the things we have to do, oddly enough, is we have to go all the way back to the beginning of everything, which was a long, long, long time ago. Okay. And at that time, it's thought that everything in the universe was concentrated into a single point, and that was called a singularity. Okay. All the stuff was in there. All right. And that led to an unstable situation that caused a giant explosion. And you probably know that the name of that explosion is the Big Bang. Okay. The Big Bang. Okay. And when the Big Bang happened, all the stuff in the universe was created. Well, what is the universe made of? It's only made of three things: matter, which is up here, okay, energy, and empty space. That's the whole universe, which is pretty amazing when you think about it, because the universe is infinitely large. Okay. Um, Matter is anything that occupies space and has mass, just like it says above. Energy is the ability to do work, and energy comes in many forms. There's chemical energy, there's electromagnetic energy, there's kinetic energy, there's potential energy, um, there's energy that comes from um, the production of heat. Okay, you can convert one energy type into another, and that happens all the time. And every time you do that, you lose a little bit of energy as heat, and that goes off into the universe and makes the universe more random, or a chemist would say it increases the entropy, the disorder. Okay, empty space. All right. When that happened, two laws went into effect, and these are called the first and second laws of thermodynamics. Well, what are those? Well, the first law of thermodynamics says that matter and energy can't be created or destroyed. So it's another way of saying there's only so much stuff in the universe, and that's all you get. You can't make something disappear, and you can't make something appear out of nowhere. Okay. The second law states that any system seeks the low energy state. Now, that second law might be a little confusing. Seeks the low energy state. Thank you. That second one might be a little confusing. What do I mean by a system? Well, a system could be anything. It can be the dust on a table. It could be the air in a room. It could be the a, a glass of water with some food coloring in it. You name it. Okay, there's any number of different systems, and if you don't put any energy into that system, it's going to find the least energetic way to continue to exist. Okay, another way of saying that is things take the easy way out. Okay, they like to be chill. All right. Matter can be a solid, a liquid, or a gas. All right. Um, the solids are composed of building blocks called atoms. We'll get into what that's all about in a second. Um, and solids don't change their shape over time and space. Okay. A liquid is capable of forming to the shape of its container, and it has a little bit more kinetic energy than a solid does. And a gas has even more kinetic energy and touches the walls of the container that it's in, such as the air in the room that you're in right now is touching all the walls. Okay. So that's even more kinetic energy. You can go from one phase of matter to another by changing things like temperature and pressure. Okay. All right. Atoms are the smallest particle of an element that maintain the chemical properties of that element. Now, this is a very old concept. It comes from an ancient Greek philosopher by the name of Democritus. And he was staring at a boulder one day, and he hypothesized that if he were looking at that boulder and split that boulder into two, right, he would have two smaller boulders, but they would still have the properties of the original boulder, right? The same texture, the same hardness, and so on, right? And then, he could do that again, get four even smaller boulders, but they would still have the properties of the original. Okay. And what he figured was that he would eventually get to a point where he couldn't do that anymore. There would be a particle so tiny that you couldn't split it and maintain the properties of the original object. And he called that particle the atom, right there. It goes all the way back to the ancient Greeks. And that's the working definition, really, of the atom we have today. It's the smallest particle of an element that still maintains the chemical properties of that element, meaning that it makes and breaks chemical bonds the same way a larger amount of that element would do. Okay. Can you split the atom? Yeah, you can. Okay. But what you get as a result of doing that is lots of energy and subatomic particles, and a lot of things happen that we can't quite explain fully. Okay. A chemical compound is just different elements connected to each other, atoms of different elements connected to each other. A molecule is atoms connected to each other. The atoms could be either the same or different. Okay. So all compounds are molecules, but not all molecules are compounds. A chemical formula is written using the chemical symbol, right? And the number of atoms that are present in that compound, right? So NaCl, which is sodium chloride, or H2O, which is water, right? The two by the H means there's two hydrogen atoms connected to the oxygen atom. Okay. All right. The elements in living organisms are listed here. Okay. The most predominant ones in human systems: C, H, O, E, N, S, right? For carbon, hydrogen, oxygen, phosphorus, and nitrogen, and sulfur. Okay. And you can see them listed up top. Okay. Most abundant. The others are present in smaller amounts, but that doesn't mean they're unimportant. Okay. Iron would be an example of something that's present, but not in huge amounts in the human body. But if I removed all the iron from your body at once, you would die. Okay. Because you would be able to carry oxygen in your blood and you wouldn't be able to make ATP. Right. This is the periodic table of the elements. You might say, well, what is that? That's all the elements in the universe on one piece of paper, which is pretty amazing when you think that the universe is infinitely large. And you'll notice in this periodic table different colors. Okay, the stuff in silver and copper and sort of yellowish gold there, and in green, right? Those are metals. Okay, metals. Okay. So basically everything to the left of this staircase. Everything to the right of that staircase is a non-metal. Okay. The heaviest stuff in the periodic table is in the lower right. The lightest stuff is in the upper left. Okay. All right. The symbol is inside each box, and above each symbol is what's called the atomic number. Okay. The atomic number is the number of protons in the nucleus of the atom. Okay. Atomic number is equal to number of protons which have a positive electrical charge in the nucleus. And the nucleus of the atom is where most of the mass is. Okay. The number underneath the symbol at the very bottom of the box, okay, is the atomic mass, right? The atomic mass equals the number of protons plus neutrons in the nucleus. Okay? The neutrons have about the same mass as the protons do. And if you look closely at those numbers, you'll see that they're not round numbers, right? They're going to have digits after the decimal point. So you might wonder, well, what's up with that? Aren't all atoms the same of this particular element? And the answer will be no. When the universe was created, some of the atoms got extra neutrons in them, and so they're a little bit heavier, but they still chemically interact with the other atoms the same way that the most abundant version of that atom would do. Okay. So these heavier forms of the atom are called isotopes. All right. Okay. When metals react with metals, they form metallic bonds and they make alloys, right? Which is beyond the scope of this course. Okay. When metals react with nonmetals, they form ionic bonds where the metal gives up electrons, which are negatively charged particles outside the nucleus, and hands them to the nonmetal, which takes on a negative charge, and the opposite charges attract each other. Okay. When nonmetals react with each other, they form covalent bonds in which pairs of electrons are shared between the atoms, and they're very, very strong. Okay. The last row here, right, the very last row, does not do any chemistry at all. They're found in their pure states in nature. So why is that? The reason that's the way they are is because they're in the ultimate low energy state. They don't have to give or take or share electrons with anybody. They just hang, okay? And they're called the noble gases, right? The numbers at the top of each column indicate the number of electrons on the surface of the atom, okay? So everybody in the 1A column over here has one electron on the surface. Everybody in the 2A has two. Everybody in the 3A has three, and so on. And these guys have what we call a full outer shell with two for helium because it's a light element, but the rest 8, okay? Which means they don't need to do any chemistry, all right? So ultimately what drives all the chemical reactions in the universe is the effort on the part of all these other elements to be like these noble gases, right? They want to fill their outer shells by sharing or gaining or losing electrons, and that makes them stable and low energy, okay? So that's the basics of what's going on here, all right? The atomic model posits that there's a nucleus with protons and neutrons in it. The protons are positively charged, the neutrons have no charge, and the electrons are negatively charged and move at the fastest speed anything can move, which is the speed of light. Okay. Move at the speed of light. Okay. The electrons are found outside the nucleus. The neutrons and protons are within the nucleus. Okay. There's our rundown on the atomic weight, the atomic number. The orbitals or shells are regions where you're most likely to find these electrons. Okay. And they can only exist there unless they're moving between shells, but they can't sit there between shells indefinitely. Okay. You can move an electron from one shell to another by adding some energy to it. Okay. But it won't be between shells. It'll be in the higher energy shell if it can make the jump. And when it does that, it'll give off some energy when making that transition. Okay. The electrons on the surface of the atom are called the valence electrons. Okay. The valence electrons. Okay. And they do the bonding because they have the most energy because of their location. Okay. They have lots of what we would call potential energy. High potential energy. Okay. So let's look a little bit deeper into the atom.
An atom is the smallest particle of an element that retains the properties of that element. Atoms are composed of smaller particles called protons, neutrons, and electrons. The central nucleus contains protons and neutrons, while electrons move constantly around the nucleus in concentric regions called electron shells or energy levels. A proton has a single positive electrical charge, and an electron has an opposite negative charge. Neutrons are neutral with no electrical charge. Each element is uniquely defined by the number of protons in an atom of that element. For example, only carbon atoms have 6 protons, and only potassium atoms have 19 protons. The atomic number is the number of protons in an atom of a particular element. Number of protons equals the number of electrons, making atoms electrically neutral. Electrons are so small that they have almost no weight. The atomic mass is the sum of an atom's protons and neutrons. The number of neutrons in a particular atom, and thus atomic mass, can vary. Atoms that have the same atomic number but different atomic weights are called isotopes. Electrons occupy electron shells in fixed numbers. The first shell, closest to the nucleus, holds a maximum of 2 electrons. The second shell holds 8 electrons, and the third shell can hold up to 18 electrons. Subsequent shells can hold higher numbers. The outermost shell, known as the valence shell, holds a maximum of 8 electrons. The electrons in the valence shell determine an atom's chemical bonding properties. Atoms such as helium, whose outermost electron shells are filled, have stable structures and are chemically inactive. Atoms with partially filled valence shells tend to lose, gain, or share electrons to either empty or fill their outer shells, producing a stable number of valence electrons. The number and kind of bonds an atom will form depend on the number of electrons needed to fill the valence shell.
So if we look at atomic structure, right, this is what's called the planetary model, right? You see a positively charged nucleus, a negatively charged electron, okay? And you can see some subsequent drawings for things like hydrogen and carbon and nitrogen and oxygen. And you'll note that in each case, their shells are not completely filled, okay? Sodium with 1, magnesium with 2, and so on, okay? Now, generally speaking, right, see protons in blue, neutrons red, electrons deep red. Generally speaking, students write this down and say, okay, that's the atom, all right? But I'm here to tell you, right, that this doesn't make sense. Okay. And why is that? Well, what do opposite charges do? They attract each other, right? So the hydrogen atom should collapse, and it doesn't. Why? And you'll love this answer. We've spent decades on this, okay? And the answer is we don't know, okay? You might say, well, that's not very satisfying, but that's kind of how science is, right? You don't always know everything all at once. You make discoveries and then you incorporate those into models like the one you see here for the atom. And then as you make new discoveries, you put those new bits of information into the model, or sometimes you have to take the model and throw it away. Okay. A model just represents what we know right now. All right. And a model by its very nature is incomplete. All right. And as we learn more, the models get better. All right. They more accurately reflect the world around us. But will they ever fully reflect it? Likely not. There will likely be some findings somehow, someday that will maybe turn some of these models on their head or modify them to a great extent. Okay. So this is one of the things you have to understand about a lot of science, right? Science that does modeling is by its very nature incomplete, right? In science, nothing is ever true, okay? It just hasn't been disproven yet, all right? And I know that's not real satisfying either, but you can show things to be false, okay, through something called the scientific method that we'll talk about in a little bit, okay? So, you know, take that with a grain of salt. There are other models for the atom other than the planetary one. Some of the models use zones where the electrons would be, and some of them are shaped like spheres, and some are shaped like dumbbells, and some are shaped like dumbbells with rings around the middle. And you know, who's right? Well, you know, we don't really know yet. We're working on it. Okay, a lot of work goes into this. This is why we have super colliders that try to smack the atom into little tiny bits and make sense out of what comes out of that. Okay, and try to use that information to make a better model of the atom. Okay, so what you do with a model is you take what the model gives you and see how far you can go. Okay, electron shells are energy levels around the nucleus with electrons in them. The first shell, number of shells depends on the atomic number. The first shell can only hold 2 electrons, the next shell 8, the next shell up to 18, but is okay with 8. Okay. So that's just the result of the mathematics to generate the model. Okay. The electrons on the surface of the atom have the most energy, and they're the ones that are involved in bonding. Okay. Electrons in more distant shells have higher energy. Shells always fill sequentially from the inside. 2 electrons in the first, and we move up to the second shell. We can throw in a total of 8, and up to the third shell, we could go up to 18, and so on. Okay. When the outermost shell is filled, the atom is stable, meaning it's low energy. Okay. Valence electrons are those on the surface of the atom, and they participate in bonding because they have the most potential energy. Okay. They determine what kind of chemical bond the atom can form. If the outermost shell is filled, the atom is inert, meaning it doesn't do any chemistry.
Okay. After 500 feet, turn left and then turn slightly left.
Isotopes
Now turn
are atoms
left and
with
then turn slightly
extra
left.
neutrons in the nucleus. Hold on a second. Hold on a second. I've got the mic on. I don't see him. Okay, so I went away. Cool. Isotopes, as we talked about, are just atoms with extra neutrons in the nucleus, which add to the mass but do not change the chemistry. Okay. Examples include isotopes of hydrogen, such as deuterium with 1 proton and 1 neutron, and tritium with one proton and 2 neutrons. Okay, so you can see that here. Okay, the chemistry is no different. Chemistry is the same. Okay. Now you might wonder, okay, isotopes. Okay, fine. That's what those are. Why do we care about isotopes? That's always a good question to ask, right? Why do we care? Well, the main reason we care in medicine is because some isotopes give off energy we can use in clinical procedures. Some examples: X-rays, right? Given off by radioactive isotopes. Let's just visualize bone without having to open you up. The X-rays expose photographic film where they go right through. The film is going to be dark, and where they're blocked, like by the bone, which is dense, there's going to be light. So it lets us see things like broken bones. Radiation therapy for cancer, right? We use radiation given off by radioactive isotopes to try to kill cancer cells. Sometimes it works, sometimes it doesn't. Okay. Another example is PET scan. Okay. PET scan, where we give you a radioactive compound, usually like glucose, and then we visualize where it goes in the brain so we can look for abnormalities in the brain. Okay. So those are some of the applications, right? The other thing, of course, being aware of the isotopes helps us with is, setting up chemical reactions to generate a certain amount of product in a way that makes sense. Remember that atomic mass we looked at back in the periodic table, that number can be interpreted as the number of grams per mole of that particular element, okay? You might say, okay, grams I get. What is a mole? A mole is just a count, okay? And a mole is Avogadro's number of whatever we're talking about. What is Avogadro's number? It's 6.02 times 10 to the 23rd, okay? Okay, so a mole of carbon weighs 12.01 grams, but how many atoms of carbon are in there? And it's 6.02 times 10 to the 23rd, okay? So you have to compensate for you have to know that there are isotopes in there. You have to use the atomic mass that's in the periodic table in order to weigh out that much and use it in a chemical reaction. Okay. All right. Ions are just electrically charged atoms, molecules, or particles. They can be positive or negative. If they're positively charged, they're called cations. And I remember that because the T here, it's like a plus sign. That's how I remember it. And anions, which are negative, right? Electrolytes are free ions present in a solvent like water. Okay, now, why do we care? Why do we care? Okay, electrolytes conduct current. Right? So that's one way in the body that we can do work, right? A moving charged particle is a form of energy called current that we can use to do work like transport, making nerve signals, making muscles contract, and so on, right? And for most cells in the body, it's also critical for maintaining something called membrane potential, okay? In which potential energy, like in a battery, say, is stored in the membrane by separating positive and negative charges across the membrane, where the outside of the cell membrane is positive and the inside is negative. Okay. And when we open a door and let the current flow, we can use that to do work. Okay. The positives will meet the negatives and the negatives will meet the positives. Okay. All right. So we got that. What's the other big thing about electrolytes? Electrolytes draw water to them. Okay. So where the electrolytes go, the water follows. The main one there is sodium. Okay. So if there's a sodium solution somewhere and there's a semi-permeable barrier, and then there's, say, pure water on the other side, the pure water will chase the sodium across the barrier until the concentration of free water molecules is the same on either side of the barrier. Okay. or until opposing forces basically result in no net movement. Okay. So those are some of the ions, cations and anions, right? Notice that everything on the left there is a metal, everything on the right there is a non-metal. Okay. Chemical bonds are just connections between atoms, okay? Hookups, and that's really all chemistry is, right? You've got the making and breaking of chemical bonds. That's chemistry, period, okay? Molecules form by chemical bonding between valence electrons because they have the most energy associated with them. Compounds are made from atoms of different elements. Molecules are connections between either identical atoms or different atoms, right? So all compounds are molecules, but not all molecules are compounds, okay? So let's take a look at chemical bonding.
When forming compounds, atoms tend to gain, lose, or share electrons to achieve a stable noble gas electron configuration. 10 electrons. Ionic is formed by transferring one or more valence electrons from one atom to another. Is formed by sharing valence electrons between atoms. Sodium has one valence electron and fluorine has 7 valence electrons. If the one valence electron in sodium is transferred to fluorine, both atoms achieve an octet of electrons. By losing one electron, sodium has a +1 charge. By gaining one electron, fluorine has a -1 charge. An ionic bond is the electrostatic force that holds ions together in ionic compounds. Both fluorine atoms have 7 valence electrons. If one electron from each fluorine atom is shared with the other atom, then both fluorine atoms achieve an octet of electrons. This type of bond is called a nonpolar covalent bond. The sharing of electrons between atoms is exactly equal, and the electron density is symmetrical. According to the colors of the rainbow, the electron-rich region is shown in red. In this case, the electron-poor region is blue. Hydrogen has one valence electron and fluorine has 7 valence electrons. If the one valence electron of hydrogen is shared with 7 valence electrons in fluorine, fluorine achieves an octet of electrons. We note that hydrogen has only 2 electrons, which is the electron configuration of helium. When bonded to another element, hydrogen will have 2 electrons in its valence shell. In this bond, fluorine has a greater attraction for the electrons in the bond, electronegativity. As the electrons spend more time near the fluorine atom, the electron density shifts towards fluorine. The most electron-rich region is red. The most electron-poor region is blue. The result is a greater electron density near fluorine, a partial negative charge, and a correspondingly lower electron density near hydrogen, a partial positive charge. This type of bond, where bonding electrons are unequally shared, is called a polar covalent bond. A polar covalent bond can be thought of as an intermediate between a nonpolar covalent bond and an ionic bond.
So when we're talking about covalent bonds, we're talking about interactions between nonmetals, the elements to the right of that staircase in the periodic table. They share pairs of electrons. In a nonpolar covalent bond, the sharing is equal, okay? So when does that occur? That occurs when the atoms are identical, okay? Or between carbon and hydrogen, right? So nonpolar covalent bond, carbon, hydrogen, okay? Or identical atoms. We should say identical nonmetals. Okay. Nonpolar compounds like to associate with nonpolar compounds because of the second law of thermodynamics, low energy. That's the easy way to arrange things, right? In a polar covalent bond, you have unequal sharing of electrons. The electrons spend more time with one atom than the other, and the result is that the ends of the molecule have slight charges, okay? So we could do an example like with water, okay? So these are covalent bonds, right? But the water has a slight negative charge on the oxygen because the oxygen is electronegative and The hydrogens, as a result, have a slight positive charge. And what does that mean? That means it likes to be with things that either have an outright charge or have polar covalent bonds in them. All right. So we can look at an example, right? Let's say that you went to the grocery store and you wanted some Italian salad dressing, right? And the Italian salad dressing, as you know, on the shelf, right? It's a terrible drawing, but hey, okay. You've got the water on the bottom. So blue there. Okay, you got the water on the bottom, right? And on the top, you've got the oil, yellow, right? Oil, right? If you shake that up, you'll make a temporary emulsion, right? The oil and the water will kind of associate in a sort of a weird way, all right? And then if you sit that bottle back down, what's going to happen? It's going to separate again, go back to its low energy state. The shaking of the bottle was you adding energy to the bottle, okay? In that case, kinetic energy, right? But then we went back to a low energy state when the energy input stopped. The oil got with the oil because the oil is nonpolar, and everything else got with everything else because it's either charged or polar. Okay. So that's the bottom line there, right? The take home is that like, oh, that's yellow. Like dissolves like. Okay. The two things have similar chemical properties. It's easy to mix them and form a solution. Okay. So there's an example. Covalent bonds can be single or double or triple, all right? There you got water, single covalent bonds. And then on the left there, you've got a hydrogen molecule, right? Note that this is a molecule and not a compound, right? Well, this is a molecule and a compound. Now, let's look at this for a minute, right? Let's do the bookkeeping here with the electrons. Here, you've got 2 electrons for this hydrogen atom and 2 for this hydrogen atom. You get to count them twice because they're being shared, right? Well, that's a full outer shell like helium, which is at the very top of the noble gases, right? How about over here, water? Well, 2, 4, 6, 8, full outer shell. So oxygen's happy, oxygen's stable. How about hydrogen? Well, 2 for that hydrogen and 2 for that hydrogen, and that's a full outer shell. So that's a stable compound. That's low energy. That's the second law of thermodynamics, okay? Single covalent bond, double covalent bond, triple covalent bond. It shouldn't surprise you that a triple covalent bond is stronger than a double, which is stronger than a single, okay? The transfer of valence electrons from one atom to another is an ionic bond, okay? The metal loses the electrons to the nonmetal. The metal takes on a positive charge. The nonmetal takes on a negative charge. Now, how do they arrange themselves? Well, kind of like this, right? This is the formation of sodium chloride, which is table salt, right? And look how the negatives are surrounded by the positives and the positives are surrounded by the negatives. Why? Well, that crystal represents the low energy state, right? Everybody's attracted to everybody, so everybody hugs everybody, so that's easy, right? If I tried to force the chloride to be on one side of the crystal and the sodium to be on the other side, I'd have to use lots of energy to do that, okay? Now, go into this a little bit deeper, right? The sodium has 1 electron in its outer shell. It donates it to chlorine, which has 7. That makes the chloride ion have 8. And then the shell underneath the sodium has 8, right? So that qualifies as a couple of stable ions. The sodium becomes a sodium cation, and the chloride is the chloride anion, right? Now, if I were to tell you that I was bringing sodium metal into school, right, I'd probably be arrested, all right? I have to bring it in under kerosene because it reacts so violently with water that it bursts into flames, okay? It's really cool to watch. You can Google sodium and water and see what happens, all right? Chlorine, I don't know if you've ever been around a pool when it's being chlorinated, but chlorine will bleach your skin and your clothes and is very caustic, right? But when you put them together, what do you make? You make table salt, which you put on your French fries. So why is table salt safe to eat, but sodium and chloride so dangerous? And the answer is you've changed where the electrons are. And as a result, you've changed the properties of both of the atoms, right? The compound now has different properties. Hydrogen bonds are interactions between partially positive hydrogen atoms that are involved in a polar covalent bond and partially negatively charged electronegative atoms that have a slight negative charge. So it's sort of a, you know, a temporary attraction, right? As they flow past each other, they shift and move so that they can, again, you know, live with each other the easiest possible way, right? Negative near positive, positive near negative, okay? Where do we see this? We see this in a solution of water. We see this with a large molecule like DNA, the attraction between the bases of the nucleotides of DNA. And it always involves a hydrogen atom attracted to an oxygen or a nitrogen atom, okay? Or a sulfur atom in some cases, all right? Let's take a look at hydrogen bonding and what that's all about.
Are in a constant tug of war with electrons. Oxygen exerts a far stronger pull on the shared electrons than does hydrogen, and so the electrons spend more of their time closer to the oxygen atom. Because of this unequal sharing of electrons, the oxygen atom in a water molecule actually has a slight negative charge, and each hydrogen atom has a slight positive charge, even though the water molecule as a whole is neutral. Because of the unequal sharing of electrons and the resulting positive and negative poles, a water molecule is said to be polar. The polarity of water molecules causes them to be attracted to each other. Since the positively charged atom involved in this special type of attraction is always a hydrogen atom, this kind of bond between molecules is called a hydrogen bond. Each water molecule can hydrogen bond to 4 other water molecules. The hydrogen bond is weak and lasts only a tiny fraction of a second, but it takes a lot of energy to overcome the combined attraction of many hydrogen bonds. This explains water's great capacity to store heat, its high boiling point, surface tension, and several other unusual properties.
So you can see here an example, right? This is an illustration of hydrogen bonding between water molecules where the positive, partial positive part of one water molecule is near the partial negative of another. They're temporary attractions, but the sum total of those attractions can be very powerful, all right? Examples of this at work. Well, when water freezes, all right, we have to maintain some spacing so that negative is near positive and positive is near negative. And what that does is that when water becomes ice, it becomes less dense than liquid water, right? So the ice will expand, right? If you've ever been like me and forgotten a 6-pack of Coke in the trunk of your car in January, right? You go out the next morning, you open the trunk, what do you have? Well, in all likelihood, is 6 split cans because the water in the Coke froze, expanded, and split the can open. This is also why cracks in your basement get bigger in the winter because the water melts and freezes and melts and freezes. And as it freezes, it expands, and that makes the crack bigger, okay? Other applications, right? Hydrogen bonding is responsible for something called capillary action. When you spill water on the table, you get a paper towel and you mop it up. Well, the paper towel is made of something called cellulose. Cellulose, as we're going to see in a bit, is a long chain carbohydrate, okay? It's a polysaccharide, and it is a polar molecule because it has oxygens and carbons and hydrogens in it, right? So the water is attracted to the cellulose in the paper towel, and the water that's still there on the spill is attracted to the water that's getting wicked up into the paper towel, and as a result, you wick up the whole spill, right? This is the same way that plants are able to get water from the water table in the soil up to the very tops of tall trees because the plant is basically made of wood, okay? Wood and other things, but the wood is what conducts the water up to the top. That's why we can have tall trees, okay? Van der Waals forces are the weakest, okay? They're dependent on interatomic distance, and they determine final molecular arrangement. Examples include antibody antigen complexes, okay? They explain how a spider can hang upside down from a ceiling and why a gecko can hang from one toe on to form from a glass surface, okay? A chemical reaction is basically the making and breaking of chemical bonds. The reactants are going to bump into each other. Bonds are going to be broken, new bonds are going to be made, and the product will be spit out, okay? Examples of chemical reactions include synthesis reactions, such as dehydration synthesis reactions, the formation of larger molecules from tinier ones. And it involves the removal of water. That's what dehydration means. And hydrolysis, where we use water to actually split a bond open, okay? So you can see examples here, right? Dehydration synthesis on the top and hydrolysis on the bottom. When you're doing synthesizing, right, you're always consuming energy because that's not something that happens naturally, all right? You have to draw on an energy source from some other place, and we're going to find out that that energy source is a molecule called ATP in living systems, okay? With hydrolysis, you release energy when you crack the bonds open, and then you can capture that energy to do other things, okay, such as make ATP, say, okay? Endergonic reactions need energy to go. Exergonic reactions give energy off, right? So the exergonic reactions supply the energy for the endergonic reactions. This is why, for instance, we have to eat to live, okay? Because the energy in the chemical bonds of the food that we eat gets trapped, gets used to make ATP, and then that ATP is used to do the work of keeping you alive and building new molecules and all the other stuff that requires energy, okay? Now, if we draw an analogy, right? Why can't we use the energy from our nutrients directly to drive the endergonic reactions? And the answer is that the cell isn't set up to understand that very well, okay? For sure, there's chemical energy in those bonds, but there is no mechanism to take that energy directly to do work. Instead, we have to steal it from the nutrient and then lose a little heat along the way. And then eventually we synthesize the ATP, which is ready to power all the reactions in the cell. Now, we can draw an analogy with your car, right? Your car, all right, runs on gasoline unless you have a hybrid or an electric, okay? And what happens when you put your key in the ignition and turn it and push your accelerator pedal down is that the gas in the tank gets fed into your carburetor, which mixes it with air, and then that mixture is sprayed into the engine block, which contains pistons and a spark plug. When the spark plug generates a spark, that mixture explodes, right? And that moves pistons that are inside the engine block, and that turns a crankshaft, and that eventually turns the wheels of your car, okay? So that's how that's taking a favorable reaction, exploding gasoline, right? And an unfavorable situation, which is your car moving down the road or say up a hill, all right? Where does gasoline come from? It comes from crude oil, all right? If we were to put crude oil in your gas tank, your car would not run, okay? It would just look at you and say, I don't know what this is. So what we do with crude oil is we crack it, all right? We break it into tinier pieces, all right? And basically crude oil is long hydrocarbons, and the piece that's in gasoline that's good for your engine is called octane. It is an 8-carbon molecule, octo for 8, like octopus, okay? It has 8 arms. And, that's why high octane fuel makes your car go far, all right? That's the process. You have to turn the energy into something that system understands in order for it to be immediately used, right? And of course, heat is given off too, okay? In an oxidation reduction reaction, what we have is the transfer of electrons from one atom or molecule to another, okay? The molecule that gets reduced, which receives the electrons, gains energy, and the molecule that gets oxidized loses energy. Now, why is that? Well, because you've lost a particle that contains potential energy because of its position, right? And you've given it to something else. And as a result, you've lost some energy, all right? So a fully oxidized molecule has very little chemical potential energy, while a highly reduced one has lots of chemical potential energy, okay? For living systems, the money bond, the one that has the most energy in it, is the carbon-hydrogen bond, all right? So molecules with lots of carbon-hydrogen bonds have lots of chemical potential energy, which means that they can provide a lot of fuel by giving a lot of calories to the system so that it can do work and do synthesis and everything else that's required for life, okay? All right, chemical notations, right? Shows the chemical compounds and reactions. The abbreviation of an element represents one atom of that element and is the chemical symbol. The number before the chemical symbol is the number of atoms. The number in the lower right hand of the chemical symbol is the number of those atoms that are in the molecule, okay? So one is the designation of the number of molecules, and the other number is the designation of the number of atoms in the molecule, okay? A superscript of plus or minus indicates that there's an ion. If it's a plus, it's a cation. If it's a minus, it's an anion, okay? Chemical reactions don't form or destroy atoms. They just rearrange them into new combinations. We change how the bonds are set up, right? We make and break chemical bonds, okay? When we come back, we're going to talk about inorganic and organic compounds, and we're going to talk about the molecules that are critical for life, okay? So at this point, we'll take 20 minutes, and we'll be back at, well, that'd be 3:53. So let's just call it 4. We'll be back at 4 o'clock, okay? So let's pull this down. Inorganic compounds do not have carbon and hydrogen in the same molecule, okay? Carbon is the basis of life because carbon can do something that no other element in the periodic table can do, which is form rings and chains with itself, which means you can build a lot of interesting tools using carbon as your basis. It's like the ultimate Lego piece, okay? Carbon dioxide and carbon monoxide, even though they contain carbon, are not considered organic compounds, okay? Acids and bases control the pH of a solution, okay? The pH of a solution is basically the acidity of it, all right? Acids are substances that lose protons in water, and bases are substances that pick up protons in water, okay? When we look at the pH scale, neutrality is 7, and anything below 7 is acidic. Anything above 7 is basic, okay? The scale runs from 0 to 14, but it's a 10-fold scale. So let me explain why that is, okay? So pull up a pen. Okay. So we're talking about pH. pH has a definite definition. pH is equal to the opposite of the log base 10 of the molar concentration of protons. Everybody whose head that went over. Molar concentration is equal to moles per liter of whatever we're talking about. All right? Liter is a volume measurement. What are moles? Mole is a count, okay? It's basically Avogadro's number of whatever we're referring to. So that number is 6.02 times 10 to the 23rd. All right. Right. So a mole of carbon, all right, would have 6.02 times 10 to the 23rd atoms of carbon in it. All right. So this is what we mean by molarity. It's a concentration. Now, if we take a solution that is, say, 10 to the minus 3 molar in protons, and big M means molar. Okay. All right. Then what is the pH of that solution? What is the pH? All right, so some stuff, size, space. And minus 3 is our concentration question. We can plug this into the equation that I just showed you. Right? So if we do that, right, the pH would equal the opposite of the log of 10 to the minus 3. Okay, well, the log of 10 to the minus 3 is negative 3. Right? The opposite of negative 3 is 3. Okay? So something that's 10 to the minus 3 molar protons is a pH of 3. Another important equation to remember is that pH plus pOH equals 14. Okay, you might say, well, what's pOH? It's the molar concentration of hydroxyl ions in the solution. Okay, so with our pH 3 solution, what would our pOH be? And the answer would be 11. Okay, so you can see that the scale varies by tenfold. Okay, if you have something, for instance, that's pH 3, right? That's going to be 100 times more acidic than something that's pH 5, because this is a tenfold difference and this is a tenfold difference. So you have to multiply that. Okay. Something that is pH 8, right, is 10 times more alkaline than a neutral solution. Okay. So you can see some of the different solutions and some of the different pHs that are listed up here, and some of the different pH ranges that different things like to live in. Okay, and the reason for this, we're going to see in a little bit, is that the tools of life, which are proteins, are selected to work at these pH ranges. All right, they do their best work in the range that the organism is normally living in. Okay, that's called the optimal pH. The same goes for temperature and goes for ion concentration. Okay, there's an optimal where the tool likes to work best. Okay, in the human body, it's 37 degrees centigrade and a pH between 7.35 and 7.45. Okay. But in other organisms, it's different. A buffer is a way to control pH. Okay, buffers convert strong acids or bases into weak ones. So if you throw a strong acid into a buffered solution, it will convert it into a weak acid, which won't change the pH very much. If you were to throw a strong acid into pure water, the pH would go down very quickly. Okay, the same for a base, right? You throw a strong base into a buffered solution, the pH won't change very much because it'll be converted to a weak base. Throw a strong base into water, the pH will rise dramatically. Okay. A salt is just material that dissociates in water and doesn't release hydrogen or hydroxyl ions. It's formed by acid-base reaction of a metal, acid, and non-metal. Okay. The naming is specific. It starts with the name of the cation, followed by the name of the anion. Some different salts are shown here. Acetate, carbonate, chloride, those are anions, okay? And the cations shown over there. So an example, magnesium phosphate would be MgPO4, okay? One example. Water is critical for life because water is a fluid, right? And movement in a fluid is possible, right? Which means that reactants can bump into each other and make or break chemical bonds. Okay? Water also participates in chemical reactions like dehydration reactions, and is produced in things like dehydration reactions and is consumed in hydrolysis reactions. It has a high heat of vaporization and a high heat of fusion. Okay, which means it can give off or take in a lot of heat without changing temperature very much. And that's because of the hydrogen bonding. Okay, a lot of things dissolve in water, polar stuff and ionic stuff, like salts and polar compounds dissolve in water, so they can engage in chemistry in water, right? And it's of course a polar molecule, right? You can see here an example of a salt crystal being thrown into water. And what will happen is that the water will basically insert itself between the anions and the cations, and eventually we'll get to the point where the anions and the cations are floating free, surrounded by water molecules, all right? Once we throw enough salt in there to occupy all the water molecules, we'll get to what's called saturation, okay? And at saturation, you can't dissolve any more salt in the water or whatever it is you're throwing in there, okay? Because we used up all the water molecules. Solutions and tonicity, right? A solution could be isotonic, hypertonic, or hypotonic, okay? What do we mean by that? Well, in an isotonic solution, the water concentration inside and outside the cell is the same. So there's no net movement of water. Net movement means that the movement out and the movement in, the rates are the same, right? In a hypertonic solution, there is less water outside the cell than inside the cell. The water leaves more quickly than it comes in, and the cell will crenate. It'll shrivel up, it'll dehydrate, okay? And that often causes the cell to lose function. And in a hypotonic solution, like pure water, water moves in more quickly than it moves out, and the cell, if it doesn't have a cell wall, will rupture. We'll learn about cell walls in the next chapter. Okay. Water can form hydration spheres, which attract polar water molecules. The ions, because of their charge, do that. Hydrophilic compounds dissolve easily in water, while hydrophobic ones do not, right? Think of a hydrophilic compound like table salt or the sugar you put on your cereal every morning, right? A hydrophobic compound would be like vegetable oil. What happens when you put vegetable oil in water? It doesn't dissolve, okay? And that's because, remember, we said earlier, like dissolves like. Okay. So you can see these hydration spheres here, okay? Organic molecules contain carbon, hydrogen in the same molecule, okay? The carbon-hydrogen bond is a non-polar covalent bond. It's also the highest energy bond for living systems, okay? Carbon can form rings, chains. It's capable of doing all this bonding, unlike the other elements in the periodic table. That's why it's the basis of life, okay? Monomers are simple units of organic compounds by themselves, and a polymer are the monomer units connected together like a chain, okay? You can see here some examples of how carbon can do its thing, okay? Rings, chains, branch chains, and so forth. We should point out that this is a little bit of a misleading view because these things do have shape to them other than what you're seeing, right? This guy here, right, which is glucose, is actually shaped like an easy chair. So the ring there kind of goes like that. That's the height of the chair. And then the bottom of the chair goes like this. And then you've got the OH and the hydrogen groups hanging off, right? The oxygen here would be at the top, okay? And so on. As I go around the chain, okay? That's how they'd be set up. And the reason for that is that carbon takes on different shapes depending on how many molecules are bound to it, okay? If a carbon has 4 molecules or atoms bound to it, then it's going to be shaped like a tetrahedron. What is that? A tetrahedron is just looks like a teepee, right? This is coming out at you. And this is going into the board. That's a carbon atom. Draw that very well. and then this guy's sticking up, okay? And then let's say this is methane, so we'll just make these hydrogen atoms, right? That's the actual shape, right? Shape matters because shape dictates interactions. If it's bound to 3 other atoms, like here, okay, it's flat, like a frisbee, okay? And if it's only bound to 2 other atoms, like in CO2, it's linear, it's in a straight line, okay? And the reason for this is to minimize interactions between the groups that are attached to the carbon atom, and that's a low energy arrangement, like second law of thermodynamics, sticking its head up again, okay? So, don't be a little misled by these pictures, okay? Carbon, hydrogen, oxygen, a ratio of 2 to 1 to 2 is the carbohydrate, right? So it's CH2O and then the subscript of N, which is the number of carbons in it. So if it was a 6 carbon carbohydrate, it would be C6H12O6. Okay. Monomers are monosaccharides, single ring sugars, that include things like glucose, fructose, galactose, ribose, and deoxyribose, which is part of the backbone of DNA. Ribose is part of the backbone of RNA. Okay. Disaccharides are 2 monosaccharides connected covalently to each other. Examples include sucrose, which we get from plants, lactose, which we get from milk, and maltose, which we get from plants as well. Let's look at disaccharides.
Sometimes organisms link sugar. Sometimes organisms link sugar molecules in pairs to form disaccharides. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of 2 linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Polysaccharides are many monosaccharides linked together. Examples include starch, glycogen, cellulose. Okay, let's take a look at how these are formed.
Polysaccharides are polymers. Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
With the exception of glycogen, these are all plant products. Okay? Proteins are the tools of life. Okay, any life process involves 2 or more proteins, at least. Okay, there's 20 naturally occurring amino acids that are the backbone of proteins. They all have an amino group, a carboxyl group, and a side chain called an R group. A dipeptide is 2 amino acids joined by a peptide bond, which is between the carboxylic acid group of 1 amino acid and the amino group of another. A polypeptide is 10 or more, okay? If you look at the amino acids, here they are. They fall into 3 groups, charged, polar, and nonpolar. All right, the charged amino acids have a positive or negative charge on their R group at neutral pH. The polar amino acids have electronegative atoms in their R groups. It's like nitrogen, oxygen, and sulfur, okay? And the nonpolars have R groups that are made up primarily of carbon and hydrogen, okay? So when you attach these amino acids to each other, what happens is that the second law of thermodynamics takes over, right? The charged residues seek out the residues of the opposite charge. The polar residues seek out water or each other, and the nonpolar residues seek out each other. Okay, so let's look how protein is made.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. A hydrogen atom, and 4. A group that differs from one amino acid to another and is represented by the letter R. This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Once they're assembled, they have their secondary, the primary, secondary, and tertiary structure. Let's take a look at this.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another.
Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone exert a strong pull on electrons, giving them partial negative charges, and leaving nearby hydrogen atoms partial positive charges. These negatively and positively charged atoms are attracted to one another.
Oops, sorry.
Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone exert a strong pull on electrons, giving them partial negative charges, and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds.
Oh, come on.
Structure. A hydrogen bond with one another, or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
Lipids include triglycerides, which we see in fats or oils, which can either be saturated or unsaturated. They're made up of a glycerol backbone and fatty acids attached to them. We call these neutral fats. They don't dissolve in water, they're hydrophobic. And at room temperature, saturated fats tend to be solids, think lard or Crisco, and unsaturated fats tend to be liquids, like oils. Let's take a look.
Butter, lard, margarine, and salad. Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules.
So you can see here some examples of unsaturated and saturated fats, okay? Saturated fats have more calories. Unsaturated fats have fewer calories. Unsaturated fats are liquids at room temperature because the fatty acid tails don't stack as easily as they do when they're saturated. So this is one of our examples of how shape matters, right? Those double bonds put kinks in the chain, so the chains don't sit on each other as easily and associate with each other, right? Phospholipids are the main structural components of plasma membranes. And basically one fatty acid is stripped off and a phosphate with an amino group is added. And that is what we call the polar head, okay? The 2 fatty acids that remain are nonpolar. And what this does with regard to the plasma membrane is forms a semi-permeable barrier, which only nonpolar things can cross on their own. If you're polar or charged, you have to have a special protein gate or tunnel to go from one side of the plasma membrane to the other. Okay, so you can see how the heads are associated with the water or the cytoplasm. And again, that's the second law of thermodynamics, right? The nonpolar part of the molecule gets with the nonpolar part of other molecules, and then the charged part gets with the polar solvent, okay? Steroids include anabolic and sex steroids, mineralocorticoids, glucocorticoids, and phytosterols. They're a different type of lipid. They're mostly carbon and hydrogen, but they have a particular shape, all right? In the human body, they form bile salts and steroid hormones. And of course, cholesterol is the starting material, right? Microbes use these for things like stabilizing cell membranes and as virulence factors, weapons to produce disease, okay? Cholesterol is the precursor of steroids and vitamin D, right? Prostaglandins are made from fatty acids. They're in a variety of tissues and they're involved in things like blood clotting and inflammation, okay? Nucleic acids, of course, are the information molecules, okay? This is where the information to build the tools of life is located in packets called genes. In eukaryotes, those genes are on one of several chromosomes, which are a combination of DNA and histone proteins. In prokaryotes, they're on a chromosome that is basically a circular molecule, all right, and does not have a nuclear membrane around it, okay? DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and the building blocks are called nucleotides. The nucleotide that's used for energy currency in the cell is ATP, okay? The bases in a nucleic acid polymer can form hydrogen bonds with the neighboring bases by a process called complementary base pairing. The nucleotides are made up of a sugar, a phosphate, and a nitrogen base. The nitrogen base is the information part of the nucleotide, right? The phosphate and the pentose sugar are always the same in each of the building blocks, okay? So let's look at DNA and RNA.
RNA is the type of sugar their nucleotides contain.
Okay.
Polymers made of subunits called nucleotides. One DNA and RNA is
Come on.
DNA.
Why? Why are you doing this? Okay, hold on a second. Hold on a second. It's just arguing with me. Let's do this.
DNA and RNA are nucleic acids. Polymers made of subunits called nucleotides. One DNA and RNA is the DNA,
Hold on.
while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 nucleotides, which differ in their
Hold on. I hate this. Let's do this. Let's do this.
adenine, guanine, and cytosine.
No, just stop. 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 All right. Let's do this. All right. So let's do. All right. This better.
DNA and RNA are nucleic acid nucleotides.
Let's do this. Where we go?
DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So we asked the question then, these genes that contain all this magic information, right? How did they happen, right? How did these genes appear? And the answer is evolution and natural selection, which is a process in which any living population has differences that are the result of their genetic content, all right? And as their environment changes, some of the individuals will live long enough to breed. Some of them won't live long enough to breed. Those with greater reproductive success are going to pass their genes on to their offspring, and they'll be better fit for that new environment. All right. It is basically a trial and error process that takes many, 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 many generations. Okay. And for us, it's hard to conceive because we don't really live very long compared to the planet Earth, which has been here for billions of years. Right. We've been here for like half a second. Okay. So you have to think in terms of infinite time when you're talking about something like evolution, because that's really what you're observing here. You have a slow, steady process, but you're looking at the end result or the current result of infinite time applied to that. Okay. If you imagine, for instance, that you had the means and the time to do something, right? If you never were going to die and you had all the tools at your disposal you could possibly need, you could do anything you want. Okay. You could stack cards, playing cards from here to the moon if you had the time and the tools, right? And then to somebody that arrived later when the project was finished, they would think of it as magic, but it really isn't. It's just trial and error and a lot of time. Okay. And that's what results in the process that you see today. All right. All right. Let's take just 10 minutes and then we're going to get as far into chapter 3 as we can. Chapter 3 is kind of long. So